We talk a lot about protein intake, but measuring what actually happens to the protein inside of the body, that's a completely different challenge. And here's the surprising part. Even the most advanced labs in the world don't agree on the best way to do it, because every method has strengths, weaknesses, and in some cases, downright bizarre assumptions. Today I'm going to take you through the real tools scientists use to measure protein metabolism. And by the end, you'll understand why answering the simple question, how much protein do we need, is way more complicated than most people think. Before we talk about measurement, we need to take a quick crash course in why measuring protein is so tricky. At the molecular level, carbs and fats are simple. They're composed of the same three elements. Protein, it's a bit more complicated. It is the only macronutrient that contains nitrogen. A protein is composed of an amine group and a carboxyl group. Those two pieces together are called an amino acid, the building block of every protein. The remaining portion of a protein is the side chain, which gives every protein its unique characteristics. When protein is in the body, it must be broken down. The amine group of the amino acid, which is the portion that contains nitrogen, can be excreted from the body in several ways, but about 90% of it is excreted as urea. This becomes one of the key things researchers track when measuring protein metabolism. But here's the catch. Measuring nitrogen doesn't automatically tell you where the protein came from or what your tissues are doing. That's where all of these different measurement methods come in. Let's look at the methods that scientists actually use, and trust me, some of these sound simple until you try doing them. Method 1. Urine Urea. This is the quick and dirty approach. Since most nitrogen ends up as urea in your urine, measuring urine urea can give you a very rough estimate of protein breakdown. And I do mean rough. Hydration changes it, diet changes it, even short-term protein intake changes it, independent of tissue breakdown. It's basically like trying to measure the size of a building by weighing the dust on the sidewalk. Great for a ballpark guess, not great for precision. Method 2. 3-methylhistidine. This one's more interesting. There's a unique amino acid found almost exclusively in the skeletal muscle proteins actin and myosin. Once your body breaks it down, you can't reuse it. You just pee it out. So measuring it sounds like a great way to measure muscle breakdown, until you eat a steak. Meat and fish contain tons of 3-methylhistidine, which completely throws off the measurement. So this method only works if someone is in a tightly controlled vegetarian diet. Method 3. Arterial-Venous amino acid difference. Now we get more precise. Researchers measure amino acids going into the muscle and coming out of the muscle. The difference? That's the amino acid metabolism. The downside is it requires blood draws from both an artery and a vein across a specific muscle. This may be a little bit more than what a person bargained for. But the two most commonly used methods, the ones you see in a majority of the published studies, are nitrogen balance and isotope tracer infusion. And spoiler, one of them is old, messy, and wildly imperfect, and the other is extremely precise but extremely expensive. Let's break them down. Method 4. Nitrogen balance. In this method, you feed somebody a diet with a known amount of protein for several days to a couple of weeks. Then they have to collect all of their urine, and I mean every drop for that entire period. To measure the nitrogen, a specialized analyzer burns up the urine sample at a very high temperature and analyzes the gases coming off of it for nitrogen content. If nitrogen intake from diet is greater than nitrogen losses, then a person is in positive nitrogen balance, representing a net protein gain and general anabolic state. If intake is less than losses, then a person is in negative nitrogen balance, representing a net protein loss in a general catabolic state. If protein intake equals protein losses, then the person is in nitrogen balance and there is no whole body protein change. Simple, right? Not really. It requires a person to carry around a big urine container all day and collect all of their urine. Even tiny mistakes in urine collection can completely ruin the measurement. And nitrogen balance only tells you the net change. So if protein synthesis and protein breakdown are both high, nitrogen balance can still look neutral. Plus, it's whole body and not muscle specific. Method 5. Isotope Tracers. This is the gold standard method. Researchers infuse an amino acid, usually leucine or phenylalanine, that's been labeled with a stable isotope for a few hours. You know exactly how much tracer goes in, but frequent blood samples are required. The analysis of the blood will then tell you how much of the tracer remains in the bloodstream and can be used to calculate the rate of disappearance. The difference between the rate of appearance and the rate of disappearance over time reflects the amount that was taken up by the tissue. This will give the researchers an idea of the whole body protein synthesis, or FSR, and some information, although limited, about the degradation of FBR. This can also be done across a specific muscle through multiple muscle biopsies of the muscle of interest over the course of the infusion. A muscle biopsy sounds worse than it actually is. During a muscle biopsy, a small sample of muscle is collected under local anesthetic using a specialized needle. The participant feels some pressure in the muscle, similar to if you were to take your knuckle and press it into your thigh. The procedure is relatively quick. The initial incision is small enough that it doesn't require any sutures, and there's usually only mild soreness in the muscle after anesthetic is worn off. So studies will often collect several muscle biopsy samples throughout the infusion for more accurate measurements of protein synthesis. But here's the reality. This method requires a gas chromatograph mass spectrometer. You can find one of these for about $50,000 for a basic model, but most are going to cost you several $100,000. It also demands trained staff. Most GC mass spec models require extensive training well beyond the typical 2-hour training session when a piece of equipment is installed. The isotopes, they're also expensive and they can be hard to get. It's also going to require your participants to lie still for hours, sometimes getting multiple blood draws or multiple muscle biopsies. And you can't run it for more than about 12 hours, which means that, among other things, you can't easily study real-world eating patterns. This is why tracer studies are rare, have small sample sizes, and are very expensive, and why labs that do them often publish fewer but higher quality papers. So now you can see why measuring protein metabolism isn't trivial. Some methods are simple but crude, some are elegant but restrictive, and some are incredibly precise but require equipment that costs more than a luxury car. For these reasons, most protein research often has small sample sizes, very complex study designs, and results that don't always translate perfectly into the real world. Understanding this context helps you to read protein research with a little more informed critical eye. We've covered the major tools scientists use to measure protein metabolism, from basic urine markers to advanced isotope-infused tracer studies that would make any biochemist smile. Each technique answers part of the question, but none of them paint the complete picture on their own. If you found this breakdown helpful, hit the like button so that I'll know to make more science deep dive videos like this. Thanks for sticking with me through the biochemistry jungle. And remember, the next time someone confidently claims to know exactly how much protein you need, feel free to politely ask them which mass spectrometer they used.